All right. So now, chapter 2, which is all about complements. So page 28. 3 lines down, it says some components of the innate immune system was talked about in chapter 1. So it says that this chapter, if you go down a little bit more, this chapter discusses other components of the innate immune system, such as the complement system and other circulating effector proteins of innate immunity, including cytokines and acute phase reactants. So regulatory mechanisms of complement are finely balanced. The activation of complement is focused on the surface of invading microorganisms with limited complement deposited on normal cells and tissues. If the mechanisms that regulate this delicate balance malfunction, the complement system may cause injury to cells, tissues, and organs, such as the destruction of the kidneys and lupus or hemolytic anemias. So the complement system, what is complement? Put a star by this. Complement is a heat labile series of 18 plasma proteins, many of which are enzymes or proteinases. So it is a series of proteins, it's heat labile, and many of them are enzymes or proteinases. Collectively, these proteins are a major fraction of your beta 1 and beta 2 globulins. So if you go down, not the next paragraph, not the next, but the next, so it says the complement system. The complement system is a component of your natural immune system. So that's your innate, okay? So your innate immune system. And it is crucial for host defense from microbial infections. An interface between the innate and the adaptive immunity. So think of it as kind of like a bridge going from your innate to your adaptive, including enhancement of immunologic memory and for clearance of immune complexes from tissues and apoptotic or injured cells. So these activities are initiated in various ways through 3 pathways. So there's 3 pathways to complement. The first pathway is the classic pathway. Out beside classic pathway, write antigen antibody. And we're going to talk about the classic pathway, but some initiators are immune complexes, apoptotic cells, certain viruses, and gram-negative bacteria, and C-reactive protein. So that's what activates that pathway. So the second pathway is the alternative pathway, and antigens activate that pathway. So that would be your bacteria, your funguses, your viruses, or your tumor cells. Okay. And then the third pathway is mannose binding lectin pathway. And what that means is certain microbes that have the sugar mannose attached to them, they activate that pathway. So there's 3 pathways. the three pathways, put a star by this next paragraph, this part right here. The three pathways converge at the point of cleavage of C3 to C3b. So what that means is, let me see if there's a picture. I don't really have a picture, but what that means is no matter how it started, whether it's the alternative pathway, the classical pathway, or the lectin pathway, they all meet and converge at C3 to C3b. So we're going to watch a video on this later, but this C3, so have we talked about C3 and C4 yet, or no? Okay. So C stands for complement, okay? And what these are little proteins, okay? And we'll go, after we kind of learn about them, we'll watch this video and kind of how they initiate and how they start. But remember how I said about complement, this activates the next, it's like a domino effect. This activates this activates this, that one activates the next one, so forth, so on. And so no matter how it started, whether it be just the microbe itself or the antigen activated the antibody to start it, or whether it be this mannose binding lectin, they will all meet at C3 to C3b. And that's what these little things are. That's what I'm talking about. So they all meet there and then go forward in the same path. So it could start this way, or this way, but they all meet at a certain point and continue on. Does that make sense? Okay. So you need to know that. They all converge at C3 to C3b, which is the central event of the common final pathway, which in turn leads to the activation of the lytic complement sequence, C5 through C9, and cell destruction. And we'll talk about that in a minute. The C5 through C9 is called the membrane attack complex. And essentially what happens is the destruction, okay, the destruction of that cell. So activation of complement. Normally, complement components are present in the circulation in inactive form. So these are already in our body, these proteins, they're just inactive, okay? And what activates them, which we're going to talk about in a minute, is the alternative pathway, a microbe, okay? An antigen is what activates it. The antigen antibody response is what activates the classical pathway. And then this mannose binding lectin activates the lectin pathway. So, but it lives in our circulation in inactive form. Kind of like I talked about chickenpox before, or shingles. If you've had the chickenpox lies dormant in your body, okay, for years and years and years, until something happens and it's reactivated. It gets reactivated as shingles. Well, this is kind of the same thing. It lies dormant in your body until something activates it to make it work, okay? So under normal physiologic conditions, activation of one pathway also leads to the activation of another pathway. So it says the classical pathway. It is initiated by the bonding of the C1 complex to antibodies bound to an antigen on the surface of a bacterial cell. So you need to know that. Classic pathway is initiated by antibodies bounding to an antigen on the surface of a bacterial cell. So if you looked at this picture right here, classical pathway, here's your little IgG antibodies. Y'all see them? And then this is considered the antigen. It binds to the antibody and it activates that classical pathway. Because, I mean, think about it. Let's go back to what we just learned. That's what happens, right? It breaks through our first line of defense and it's there. Remember? So that's what happens. And it says it starts with C1. And these don't go in order, unfortunately. It's not 1, 2, 3, 4, activates 5, 6, 7. Unfortunately, it's not like that. It goes to like 1 to 4 to 2. It's crazy. But anyway, so C1 is what starts it though. The alternative pathway is initiated by contact with a foreign surface, such as a polysaccharide coating of a microorganism. So the antigen itself, it doesn't have to bind to antibody. The antigen itself is what initiates the alternative pathway. And you need to know that. The next one, the mannose binding lectin pathway is initiated by binding of the complex of mannose binding lectin and associated serine proteases to arrays of mannose groups on the surface of the bacterial cell. So what that fancy thing is saying, breaking it down, is on the surface of the antigen, it has this sugar, mannose. And if it contains that sugar, that's what activates it. Does that make sense? If it contains mannose, it activates it on the surface of the bacteria. Y'all see why we did the other chapters first? Yeah. Because we're not even into it yet, and it's already like. So if you go over to that key concept, facts about complement, you need to know all three of those. Make sure you know them. That complement is a heat labile series of 18 plasma proteins. They're present in the circulation in inactive form, and it's 3 cascades: the classic, alternative, and mannose binding lectin pathways. So enzyme activation. After complement is initially activated, each enzyme precursor is activated by the previous complement component or complex. So that's the domino effect that I was telling you. Is that after it's activated, it activates the next one. That one activates the next one. That one activates the next one. Okay. This converts to the enzyme precursor to its catalytically active form by limited proteolysis. The pathways leading to the cleavage of C3 are triggered enzyme cascades. During this activation process, a small peptide fragment is cleaved and then a membrane binding site is then exposed. Because each enzyme can activate many enzyme precursors, each step is amplified until the C3 stage. Therefore, the whole system forms an amplifying cascade. So that was what I was telling y'all about. That's the domino effect. Okay, that one activates the next one. That one activates the next one. That one activates the next one, so forth, so on. The next part about complement receptors, if you go down 4 lines, the only part that you kind of need to know in this whole thing is complement receptor 1, which is CR1, is important in enhancing phagocytosis. So let's talk about the classic pathway. All right, so the classic complement pathway is one of the major effector mechanisms of antibody-mediated immunity. The principal components of the classic pathway are C1 through C9. The sequence of component of activation is C1, then it goes to C4, then C2, then C3, then 5, 6, 7, 8, 9. C3 is present in the plasma in the largest quantities. Put a star by that. You have more C3 than anything. So C3 is present in the plasma in the largest quantities. So if you turn the page, the classic pathway has 3 major stages. Recognition, amplification of proteolytic complement cascade, and then the membrane attack complex. Out beside membrane attack complex, write C5b through C9. So recognition. The recognition unit of the complement system is the C1 complex, which is C1q, C1r, and C1s, which is an interlocking enzyme system. In the classic pathway, the first step is initiation of the pathway, and it is triggered by recognition by complement factor C1 of antigen antibody complexes on the cell surface. So what it's saying is the classical complement pathway is triggered by antigen antibody reaction. Okay. When the C1 complex interacts with aggregates of IgG with the antigen on the cell surface, the 2 C1 associated proteases, which is C1R and C1S, are then activated. put a star by this. A single IgM molecule is able to fix C1, but it takes at least two IgG molecules to fix C1. Now, why do you think that is? What is IgM versus IgG? It's bigger. It's bigger. So it has more potency to it, so to speak. Does that make sense? So remember how I told you in blood bank that IgM antibodies, we can just spin them down and they show up. But IgG antibodies, we have to warm them and add stuff to them in order for them to show up. Okay, and that's because they're smaller. They don't have as much potency to them. That makes sense? So your ABO antibodies, okay, which is A and B, right? Whenever you're searching for them, those are IgM antibodies. All you have to do is spin them in the Serology, kind of how we did. We remember we made the cell suspension, added the antibody and the antigen, spun it down, and it was there, right? Okay. But if we want to see IgG antibodies, then we have to heat them up and stuff like that, which I don't know how to do yet. Does that make sense? Because IgM is more potent. So that's all we had to do. We just had to spin them and we saw agglutination. Does that make sense? So a single IgM molecule is able to fix C1 or activate it, but it takes at least two IgG molecules. Then go down to the next paragraph. The resultant C2A fragment. So what happened is C1, it activated it. This is a horrible picture. Let me get this one. C1, so here's your IgG antibody. And see right here, here's 1, 2, 3, 4 IgG antibodies, technically, right? So IgG antibodies bound to antigen activated the classical pathway. Here's your C1. And so then it goes from C4 to C4b, and that activates C2, and then it combines with C2a. Y'all are going to learn all that. And what it's saying is the resultant fragment C2a, which is right here, C2 activated the 2a, so now you have C2a, joins with C4b to form the new C4b C2a enzyme. So here's where we are. C4b C2a enzyme, which is also known as your C3 convertase. So basically what you need to know is that C4b C2a is also the same thing as your C3 convertase. Make sure you know that. So, what we have, antigen bonded with antibody, activated the classical pathway of complement. Okay, we got that part? And it says it goes in order from C4 or C1 to C4 or C2. So it activated C1. Okay, then it went to C4. C4 then turns into C4b, which activated. So you see how right here there's like a notch missing out? This part right here made C2. Then C2 activated 2A, okay? And then these fragments right here, so now these are gone. Does that make sense? These are gone, and so now what you have now is this fragment right here and this fragment right here left. So you have C4B and 2A. They combine together to make your C3 convertase. That makes sense? And you'll see it in the video kind of how it'll put it all together. So does that make sense? This activated C1. C1 turned into C4. Now C1's gone. Okay, we don't have C1 anymore. So once it activates and then activates something else, it disappears. Does that make sense? So now you have C4. C4 combined and made C4B. Part of C4B made C2, and it made 2A. And then now you have these two left over and these are gone. That makes sense? So now these combine to form your C3 convertase. Okay? So then amplification of proteolytic complement cascade. So if you go to the next paragraph, the complement cascade reaches its full amplitude at the C3 stage, which represents the heart of the system. The C4B C2A complex, which is the classic pathway C3 convertase, activates C3. And that's really all you need to know. Activates C3. C3, you keep going a little bit more, C3 turns into C3B. And what did we say about C3B? C3 to C3B. What happens right there? They join together and create the common final pathway. Does that make sense? Yeah? Kind of. Okay. So how it is you have antigen antibody, C4, then eventually C2. Then you have their fragments left over, the C4B2A or C4B2A, same thing. That makes your C3 convertase, okay? Then your C3 is left over, and then you have C3B. Okay, makes sense? So then the membrane attack complex. The membrane attack complex is a unique system that builds up a lipophilic complex in cell membranes from several plasma proteins. Go down, not the next paragraph, but the next. So if you look right here, you have C3B, then you have the C3A going off wherever. And so then you have, eventually it goes from C3B to C5. Okay. Then C5B, it has right here. So it says the C5B, 6, 7, and 8 then form C9 to form a tubule or pore which spans the membrane of the cell being attacked, allowing ions to flow freely between the cellular interior and exterior. Complexing with C9 accelerates osmotic cytolytic reactions. So if you go up to 31, ions flow out, but large molecules stay in, causing water to flood into the cell. The consequence in a living cell is that the influx of sodium and water leads to the disruption of the osmotic balance of a cell, and it produces cell lysis. What is cell lysis? What is lysis? What's that mean? Destroys it, death. So cell death. Okay. So does that make sense? So your C5b, 6, 7, 8, and 9. So C5b through C9 is your membrane attack complex. And what that membrane attack complex does is eventually leads to cell lysis. Well, I promise we're gonna watch a video of it. It'll all come together. So it's a cascade, okay? The previous one activates the next one. That's how it works. And eventually it leads to cell lysis or cell death. So the alternative pathway. The alternative pathway shows points of similarity with the classic sequence. Both pathways generate a C3 convertase that activates C3 to provide the pivotal event in the final common pathway of both systems. However, in contrast to the classic pathway, which is initiated by the antigen-antibody reaction, the alternative complement pathway is predominantly a non-antibody initiated pathway. So it is only initiated by antigens. The antigen and the antibody do not have to bind together to activate this. An antigen does it itself. So a bacteria, a fungus, a virus, whatever. And isn't that what complement is supposed to do in your innate immune system? So if you go back to what we've learned, okay, it broke the first line of defense. Now you have your second line, which includes complement. Okay, so you're performing phagocytosis. Well, what's happening in phagocytosis? What is it doing to an antigen? Eating it and gobbling it up, and then it fuses it and makes the phagosome, right? Which is a what? Which cells perform phagocytosis? Neutrophils. Your monocyte macrophages. Also, your eosinophils can do like parasites, and then your basophils can do like allergies, so like pollen and stuff like that. So they can, but not as much. Neutrophils are your main ones. Neutrophils are your macrophages, monocyte macrophages. So what's the first step of phagocytosis? So here, let's just say this is, fat. Chemotaxis. So here's a neutrophil, right? We saw those earlier, didn't we? Okay. So you have a bacteria that enters your body, neutrophils, chemotaxis. Okay, it goes to them. It gobbles them up, right? Now, it puts them in here. So now, and then you have your phagosome formation, right? So what do you think helps with the phagosome formation and the digestion and destruction? Complement. Exactly. Complement. All this stuff that's activated, that's what causes the cell lysis, meaning the cell will be destroyed and there goes that bacteria in the cell. Does that make sense? Okay, that's why I had y'all learn the overview before you learn this part. So the alternative pathway, just an antigen. So just the bacteria or just the virus or whatever, it activates it. It does not have to bind to antibody. Okay. Microbial and mammalian cell surfaces can activate the alternative pathway in the absence of antigen-antibody complexes. Sorry about that part. Don't care about that part either. Okay, so mannose binding lectin pathway. 3 lines down. Mannose binding lectin, which is a pattern recognition molecule of the innate immune system, binds to an array of terminal mannose groups on a variety of bacteria. So once again, fancy way of saying, if it contains mannose, which is a sugar, on the surface of that bacteria, this pathway is activated. Okay. So the biological effects of complement activation. So after the bullets, the activation of complement and the products formed during the complement cascade have a variety of physiologic and cellular consequences. The effects of complement in immunity and inflammation that are mediated by proteolytic fragments generated during complement activation, such as opsonization and phagocytosis. So also, if you look, I guess back at this one or whatever, what it's saying is, these products right here that kind of merge off, so to speak, that what they do is, say it says inflammation right here. So they promote inflammation. They promote opsonization in the phagocytosis. Okay. Does that make sense? So we learned about that. Opsonization is speeding up phagocytosis, right? But it's actually these complement proteins being activated that activates that. Does that make sense? Yeah? Okay. So it says the effects of complement in immunity and inflammation are mediated by these proteolytic fragments, okay, generated during complement activation, such as opsonization and phagocytosis, and anaphylactoxins are important consequence of complement activation. These fragments may remain bound to the same cell surfaces at which complement has been activated or may be released into the blood or extracellular fluid. In either situation, active fragments mediate their effects by binding to specific receptors expressed on various types of cells, including phagocytic leukocytes and the endothelium. So what it's saying is, so we learned, go back to this one. So like, I don't know, let's just say your alternate path, or we can do the classical pathway. So this, so there's, for every one of these, I don't think I mentioned that a minute ago. So you have C4, there's C4A and then C4B, which makes the C4B, you have C2, C2A, and then those combine together. But you're like, what happened to C4A? Because you see it ain't even here, right? So those are what they kind of go off and do other things. They attach to a phagocytic cell and make them perform opsonization. So they speed up phagocytosis. Or they could attach to the endothelium and promote the swelling in inflammation or promote the redness. Does that make sense? So they, once they're activated and they go away, they're actually going and doing other stuff. Okay. Does that make sense? But, and then while they're going and doing all that, you have the C4B that's being left over to combine with the 2A to turn into C3. Does that make sense? So everybody's doing something here. Everybody's got a role. Does that make sense? Kinda? Okay. It's really hard to teach complement because you're sitting here like, yeah, I mean, I get it, I guess. You know, I don't know. It's like, it's hard to teach complement because it's hard to, videos are the best way to view complement, but we got to talk about it so you know what to look for. But
I think you're doing an awesome job.
well, thank you.
Yeah.
Thank you.
Yeah.
I actually, it made me feel so good the other day. One of my other students, we were talking about, so I have a guy in my senior class, well, I have two guys, but one of the guys in there, when I say that the whole semester, he didn't, or like the 3 semesters, he didn't talk at all, like nothing. I had to make him talk to me, and he's super intelligent, like super smart. But like even in clinical over there in the lab, like he would talk. He's the one that hit the floor. When he saw fake blood. That was him. But anyway, but he's super smart, but he's just so like very quiet. So I was kind of concerned, you know. I mean, he's smart, so I knew what he was doing. I wasn't concerned with his skills going to clinical, but let me tell you, these lab people, they like to talk, okay? And so when I was in there, or whenever I see him in the clinical, I was like, oh, kind of worried about him, you know, because he doesn't talk. So I was just like, I hope he's gonna do a good job, you know, kind of open up maybe in clinical, because they're gonna expect you to talk to them. You know, I mean, that's part of your learning experience is communication. And so anyway, me and another student, because they were just asking how everybody was doing and this, that, and the other. And then a lot of the people, he is in Alma, doing his rotation. Well, the girl that's in Douglas, they're kind of side by side. And a lot of people that are in Alma work PRN in Douglas. And so they were saying, she was saying that they had mentioned him. And I was like, well, I hope it's good, you know, because I said, I've had nothing but, like when I received, he brought me his evaluation by, and they like praised him. I mean, he did great. They literally put on his thing that he was the best student that they have had in a really long time. And I was like, oh, makes me so proud. The only bad thing they had, because there's a point, there's on your evaluations, they have to put your strengths and your weaknesses. And it's not made to, make you feel bad. That's just, they're grading you. That's what they do. That's their job. And it didn't affect his grade or anything, but they still put it down there just to make a note. And they're like, he needs to open up more. He's shy. And I'm like, well, good luck, because I tried for 3 semesters. But that's just him, But they were bragging on him and saying that he was like the best student they'd had come through. And I was like, that just makes me so proud. And I was telling that girl that, and she's like, let me read you what she said. And I was like, it made me feel so good. I said, well, his evaluation said that he's the best student they've had in a long time. I said, I'm so proud. And then she said, well, the best teacher also produces great students. So props to you. And I was like, that was so sweet. But I mean, I really do. I don't know. I love teaching, but sometimes it's just hard to teach stuff and put it into words. But with these, I mean, we need to talk about it so when you watch the video. The videos kind of put it together. But anyway. So what it's saying is these fragments can remain bound to the same cell surfaces at which complement has been activated, or either they're released in the blood or the fluid. Either situation, active fragments, and when it says C3A or C2A or whatever, that A means activated, okay? So these active fragments mediate their effects by binding to, your phagocytes or your endothelium, and they promote oxidation, or they promote inflammation, okay? So right here, you see your C4, and then you have your C4B, that's your C4A. You're like, well, where did it go, right? So C4A, which means it's activated. So that C4A, that activated fragment, will then go to the cell and activate and promote oxidation in phagocytosis, or it'll either promote swelling and inflammation. Does that make sense? Yeah? Okay. So if you go down to the next paragraph, 4 lines down, it says the activation of complement also plays a role in mediating hypersensitivity reactions. So usually that is usually when an IgE antibody, like it's like pollen comes in, and of course, what does an IgE antibody have to do with? Allergies. So everybody's allergic to pollen, right? Nobody. That is not normal. Nobody likes pollen. So pollen comes in there's your first line of defense. It gets in. Here's your second line of defense, which includes complement, right? So that pollen molecule combines with an IgE antibody, and then it'll promote the classical pathway from there. And then this C4A right here, so this is all activated, that C4A goes and promotes your itchy, watery eyes, okay, your runny nose, all that good stuff. Does that make sense? And then that's kind of how it all comes together, okay? So if you turn to the next page, in either case of activation of complement components from C3 onward leads to the generation of anaphylactoxins in the immediate hypersensitivity reaction. So it says C3-4. So if you keep going down or whatever, this part right here, if you get down to here, that, what it's saying is that's what will eventually cause anaphylaxis. Is because, yes, it's, at the same time, remember your basophils, so let's just say this was pollen with an IgE antibody. Remember, you have your basophils, which are parts, remember you have the cellular and humoral component. So in the cellular part, your basophils would be working, right? And then your humoral part, here's your complement. And so it says when you reach this C3, okay, it promotes the degranulation of the histamine and heparin, okay, and then that's when that's released and it causes the anaphylactic reaction. Does that make sense? Which is your, your throat closing up and all that. That's what causes it. It's complement. Make sense? Okay, is it all kind of coming together? Okay. In table 2.1, your classic complement pathway, the one I have highlighted for that is B lymphocyte activation. So underline that, classic complement pathway. Obviously, it says immune complex removal because that's what the point of complement is, right? But it also activates B lymphocytes, which we knew that, right? Because that's your second line of defense, which then you have your antigen and your antibody, right? Okay, that combines, and then your T cells promote your B cells, but also complements going on, which promotes more B cells, which then promotes plasma cells, which promotes even more antibodies, right? So all that's going on at the same time. The C3 to C3b is your opsonization and phagocytosis. So your C3 to C3b right here is kind of where it's going to tell it needs to opsonize phagocytosis. It speeds it up. Your C5a, your C3a, and your C4a, so those activated ones, they're responsible for inflammation and anaphylaxis. So if it has A on it, C4a, C5a, C3a, remember that's the activated component, and that's responsible for your inflammation and your anaphylaxis. It's going to go to your tissues and say, hey, you need to expand blood vessel size. So the blood vessel expands, and then what flows to there? Blood, right? Hey, you need to swell. Hey, you need to release histamine and heparin to get rid of this pollen because it ain't supposed to be here. Okay. Your C5a, it says polymorphonuclear leukocyte activation. So your C5a, which is way down here, so that's what's going to activate essentially your neutrophils. Okay, polymorphonuclear leukocytes are neutrophils. And then C5 through C9 is, or actually C5b through C9, that's your membrane attack complex, which is the lysis of the cell. That makes sense? Okay, so before we get in any, like talking about alterations and complement levels and stuff like that, we're gonna watch a video to bring it all together. Maybe. I might have just went on YouTube.
The thing that's different about a Virgo vacation home, you always have the whole place to yourself. Just you and your people. The complement system is an essential and powerful part of our immune defense system. Its main function is to coordinate the destruction and removal of infectious organisms, known as pathogens, or damaged cells in our body that need to be replaced. The complement system consists.
Let me go back to, I haven't mentioned this yet. Mainly we've been talking about pathogens, right? Old cells. How does our body get rid of them? Complement. They pretty much perform phagocytosis of old cells, and complement's activated. And so like, for example, we're gonna talk about complement even more. We have to learn what complement is, but we're really gonna talk about complement in blood bank. Because what happens is, so we've talked already about this essentially, but we just didn't bring this into it. What happens when you give, if somebody, let's go back, if somebody has the A antigen, right? They have what antibody? The B antibody, okay? Well, what if I gave them B blood, which is, if you're just giving them red cells, you're only giving them an antigen, right? So if I gave this B person A blood, this A person B blood, what would happen? Is that okay? No, what would happen? Agglutination, due to what? Same antibody, same antigen. Same antigen, same antibody, agglutination, right? So agglutination, that happens inside the body. We gave this blood to this person, right? This is inside their body, okay? Here's their arms and their legs. Just think of that. This is inside their body. This is me. Accurate. Anyway, so this is inside, inside their body. Okay, so we just gave them this. So that's what's gonna happen, is these red cells are gonna start lysing. What causes the lysis of cells? Complement. Complement is activated because antigen, antibody. So your classic complement system was just activated by you doing that. It goes through those steps, and the last step is the C5b through C9, which is cell lysis or cell death, right? And so that's what's happening inside your body. Well, when this is happening inside your body and your red cells are being destroyed, what else happens? What do you become when your red cells are destroyed? You have no red cells. What's the term for that? Anemic. You become anemic. You have your red cells carry what? What did I say earlier? Oxygen. They carry hemoglobin, which contains oxygen, right? Okay, so oxygen needs to go to the tissues in order to live. Well, if they're eating all your red cells, okay, cell lysis is happening. Complement has been activated because you just gave them the antigen and it combined with antibody. Same antigen, same antibody, not good. Okay, complement has been activated. It's taken place. Cell lysis is the end result. So it's eating all the red cells. You become anemic. There's no oxygen to your tissues. What happens when you don't have oxygen to your tissues? You die. See how it all builds on each other and comes together? Okay, so we've been talking about pathogens, okay, bad stuff, viruses, bacteria, but it doesn't have to be that. It can also be these type of antigens and antibodies. That makes sense? Or even old red cells. It doesn't, nothing has had to happen technically. Even old red cells. It gobbles them up and gets rid of them. Okay. Does that make sense? Okay.
The complement system consists of a cascade of proteins, which activate each other by cleavage, the process of splitting each protein one by one. The complement system may be activated by 3 different pathways: the alternative, classical, and lectin pathways that all run through complement C3, the central protein in the cascade. C3 serves as the master switch, controlling all downstream effectors that ultimately cause destruction of the cell. The alternative pathway is spontaneously activated through direct binding of complement to a pathogen or damaged host cell to help the body clear away debris. The classical pathway is activated when an antibody, a protein produced by our immune system, recognizes and binds to a component on the surface of a foreign cell, forming an immune complex, starting an immune response that targets the pathogen for destruction. The lectin pathway is activated by natural molecules in our body that recognize sugars at the surface of infectious organisms. In situations where the complement system becomes overactivated, it results in immune attack, the destruction of healthy cells, and ultimately causes illness to the person.
Right there, remember how I told you that, for example, people that got COVID to begin with, okay? Remember, most of the time they got COVID and then they got rid of the COVID, but they were still super sick on the vent, everything like that. This is what happened to them, essentially. Their immune system was in overdrive because it was just recognized by something that they had never seen before. And so your body did not know how to react. It went into overdrive. And this is essentially what happened.
This is illness to the person.
I'm done.
Armando Hasurigan, Biology and Medicine videos. Please make sure to subscribe, join the forming group for the latest videos. Please visit Facebook, Armando Hasurigan. Like, please. And here you can also ask questions, answer some questions, and please post some of your artworks or any artworks. You think are good. And please change the quality settings to the highest one for better graphics. In this video, we're going to talk about the complement system, and this is just going to be an overview to see how the complement proteins and the pathways are all related in some way. Now, what happens when a pathogen invades the body? When a pathogen invades the body, the first line of defense is the innate immune system, which comprises the physical barriers and the chemical and biological barriers afterwards. Now, when the pathogen successfully infiltrates our body, we have complement proteins in the bloodstream, the blood vessels. And these complement proteins are part of the innate immune system. And they are in the bloodstream because they're soluble proteins. And what these complement proteins do, they do a few things, including performing opsonization, making a membrane attack complex, and they also enhance inflammation. Overall, the complement proteins aim in destroying the invading pathogen. So what does opsonization mean? Well, opsonization is a process where the complement proteins essentially coats the outer surface of the pathogen, which allows phagocytes, in this case a macrophage, to engulf the pathogen much more easier, because the macrophage contains special receptors for specific complement proteins. Complement proteins can also make a membrane attack complex. A membrane attack complex is essentially when a group of complement proteins makes a hole in a pathogen, which causes inrushing fluids, creating a disbalance in osmolarity, which causes the pathogen to lyse, to be destroyed. And we will see later on in this video how the complement proteins initiate opsonization, the membrane attack complex, and enhance inflammation. But for now, let's see where complement proteins come from. The complement system is composed of more than 30 types of proteins.
So this says
Now, the
30,
complement
but our
proteins
book
is
says
produced
18.
by the liver,
Who
and
really
the complement
knows?
proteins will then travel through the bloodstream. Now, these complement proteins circulate in an inactive form, and so they don't attack the body. But when they come across
So we learned that, right? They're in there. Our book says 18. And they circulate in inactive form, okay? They're inactive because they need to be in there to be activated, okay? But they're inactive while we're not sick. Does that make sense?
pathogen, they will become activated. And when the complement proteins are activated, they will then opsonize, create a membrane attack complex on the pathogen, and also enhance inflammation. The complement proteins don't actually get activated that easily, because actually the complement proteins interact with each other and induce a cascade of activation of complement protein activating another complement protein.
Grammarly has saved me so much time. It double checked my writing to make sure there's no spelling errors, which can be a total lifesaver.
And so because of this, there are many complement pathways, because there's many types of ways that complement proteins can activate each other. And these complement pathways are the classical pathway, the alternative pathway, and the lectin pathway. The classical pathway being the first to be discovered. The complement proteins involved in the classical pathway is a C1q, C1r, C1s, C4, and C2. In the alternative pathway, we have factor D, factor B, properdin, and C3. And for the lectin pathway, we have MBL, mannose binding lectin, ficolin, MASP2, C4, and C2. Now, honestly, don't be intimidated by all these types of proteins. We will learn about them slowly. But what is to know about this small diagram is that the classical pathway, the alternative pathway, and the lectin pathway, all these proteins will
Where do they go? Where do they bind together?
essentially help in splitting or activating complement protein 3 to make up C3A and C3B. And it is C3A which enhances inflammation together with other complement proteins, and C3B which initiates opsonization and as well as lysis of the cell to create a membrane attack complex. So C3 is an important protein to remember, and C3 gets activated into complement 3A and complement 3B. Now, so complement proteins typically have a C in front of them. So for example, we have C1, C2, and C3 here. Complement proteins also tend to be activated when they are cleaved by something. So for example, when C3 is cleaved to C3A and C3B, these are the activated complement proteins. C2A can become activated to C2A, and C2B. C5 is cleaved to the active form of C5A and C5B. I hope you understood that. But for now, let's look at the complement pathways and see how all these
I'm gonna
pathways
post these
will
or
essentially
you can
activate
study YouTube
C3
yourself,
to
but
cleave it into
I'll
C3A
post these
and
so that
C3B.
you could. And
So
because this
remember,
really
we have
does
three
help,
complement pathways, the
um,
classical pathway, the
you can
alternative
slow it down and go
pathway,
at your
and the
own
lectin
pace
pathway.
to make notes.
Here we have a pathogen, the screen. And on the pathogen surface, we can either have, antigens, we can have mannose, carbohydrates, and also short oligosaccharides, depending if it's gram-negative, gram-positive bacteria, or whatever type of pathogen it is. Let's look at the classical pathway first. The classical pathway gets initiated or activated when antibodies bind to the antigen of the pathogen. And then when this happens, some complement proteins composed of C1q, C1s, and C1r bind to these antibodies. Well, actually, C1q binds to the Fc portion of the antibody. And essentially what this C1s, C1q, C1r complex does is that it forms another complement protein complex known as the C4b2A complex. Another name for this is the C3 convertase. Because it is a C3 convertase, it is also an enzyme. And we'll look at the function of C3 convertase later on. But for now, let's stop there and look at the lectin pathway, which is very much similar to the classical pathway, but involves different types of proteins in the activation of the C4b2A complex, the C3 convertase. Now, the lectin pathway differs to the other pathways because what initiates it are proteins binding to carbohydrates on the pathogen. So, for example, we have a protein called phylcolin, which binds to the oligosaccharides of a pathogen, a bacteria. Phylcolin also has other proteins bound to it, the MASP1 and MASP2. Another type of protein is the mannose binding lectin, or abbreviated MBL, which binds to mannose parts of the pathogen. Mannose binding lectin also comprises of other proteins, the MASP1 and MASP2, similar to the phylcolin. And MASP stands for essentially mannose-associated serine protease. So there's two types, the mannose-associated serine protease 1 and mannose-associated serine protease 2. Now, essentially, what phylcolin and mannose binding lectin do with the other mannose-associated serine protease is that they also form a complement protein complex, exactly the same as the classical one, the C4b2A complex, also known as C3 convertase. So as you can see, the classical pathway and lectin pathway involves different proteins in the initiation or the formation of C3 convertase, known as the C4b2A complex. And we skip the alternative pathway for now because the alternative pathway actually becomes activated when the classical pathway and lectin pathway forms this C4b2A complex, the C3 convertase. And we'll soon see why. So now the question is, what does the C4b2A complex do, this C3 convertase? Well, the C3 convertase is on the surface of the pathogen. And what it essentially does is that it activates the C3 protein. So the C4b2A complex splits the C3 to form C3b and C3A. Now that we've got this formation of C3b and C3A, we go back to the alternative pathway and see how the alternative pathway is initiated. The alternative pathway is initiated when the C4b2A convertase activates C3b. The C3b then binds on the surface of the pathogen. The alternative pathway will then form another type of C3 convertase, different to the ones formed by the lectin and classical pathway. This C3 convertase is called C3b-BB complex. The alternative pathway can also form another type of C3 convertase, this time with a protein called properdin. And this complex is known by the same name, C3b-BB complex, which is, as I said, a C3 convertase. So as you can see, the alternative pathway is essentially enhances the classical and lectin pathway, or the classical and lectin pathway enhances the alternative pathway. But all these pathways, the classical pathway, the alternative pathway, and the lectin pathway, all form a C3 convertase. And what a C3 convertase does is that it activates the C3, which basically splits a C3 into a C3A and a C3B. So this is the main thing to take in. Okay, so now we have the activated form of C3, C3A and C3B. Now, what do? C3A with other complement proteins, such as C5A, which is actually one of the most important complement proteins, will enhance inflammation. Actually, all the complement proteins with A will do some form of inflammatory process. So in this case, C3A and C5A will stimulate mast cells to secrete histamine. What does histamine do? Histamine enhances inflammation. Histamine increases vascular permeability, which allows leukocytes to pass through more easily. And histamine also attracts leukocytes.
On every vacation at a verbal home, there's someone like you who reunited.
And in this case, we can see a macrophage and neutrophil passing through. Now, what does C3b do? And the important thing to know is that C3b can form theoester bonds. C3b, if you remember at the beginning of the video actually initiates opsonization and initiates the formation of the membrane attack complex. Actually, important thing to know is that there are actually many C3bs and many C3as, because the classical pathway, the alternative pathway, and the lectin pathway forms many C3 convertase, which means that they split many C3s to form many C3bs and many C3as. So it's important to know. Now that we have many C3bs, these C3bs can actually bind to the pathogen surface through this theoester bond. Now, as you can see, we can see many C3bs forming theoester bonds to the pathogen surface. So it essentially coats the pathogen, a process called opsonization. And this allows macrophage to easily engulf this pathogen once it's been coated. Now, a macrophage actually has two special receptors for complement proteins, a CR1 and a C5a receptor. A macrophage cannot engulf a pathogen coated with C3b just like that. It actually needs the help of another protein, complement protein, called C5a. So when C5a complement protein binds the C5a receptor of the macrophage, this then allows the macrophage, or helps the macrophage, bind to the C3b protein through the CR1 receptors. And then through this, the macrophage can perform phagocytosis, eating up the pathogen. C3b can alternatively bind to the C3 convertase, specifically the C4b2a complex from the classical pathway of the lectin pathway, if you remember. Now, when it binds, the C3b will bind to this C4b2a complex, which will then form the C4b2a3b complex, also known as the C3-C5 convertase. And so this C3-C5 convertase can do two things. It can actually still split or activate C3 to form C3a and C3b. And now it can actually cleave and activate C5 to C5a and C5b. And remember, C5a can enhance inflammation, or it can bind to the macrophage's receptor. C5b is an important protein because it initiates the terminal stage, the formation of the membrane attack complex, abbreviated MAC. And so C5b, together with other complement proteins of higher number, such as C9, C8, and C7, will form this MAC, the membrane attack complex. And the formation of the membrane attack complex on a pathogen's surface membrane will cause the pathogen to lyse, to self-destruct, or cell bursting, destroying the pathogen. So as you can see from this overview of the complement system, the complement proteins and the system itself is very important for the body, especially in the first line of defense, in the innate immune system. Because there was simply an overview of the complement system, I'll provide links on the screen which you can click to for the classical pathway, the alternative pathway, and the lectin pathway to see in more detail. as well as a link.
So all this is going on right after essentially it enters your body. We'll watch this one real quick and then we'll watch this one tomorrow to refresh.
In this video, we will study about the complement system. We will talk about the overview, the various activation pathways involved, and the various immune effects of the complement system. Before beginning, make sure to subscribe us on YouTube for all the upcoming videos, and also make sure to like our page on Facebook for all the flashcards and interesting stuff. Links are in the description below. So the complement system is a group of about 20 plasma proteins that form essential part in the innate immunity. The word complement is used because the complement system enhances or complements our immunity. The complement system proteins are mainly synthesized in the liver and then widely distributed among the body.
You do need to know that. Write that down. Complement proteins are made in the liver. You're going to figure out that your liver and your kidneys are almost just as important, or they are as important as your heart and your brain. Complement proteins are made in your liver. So if this has to do with your immune system and enhancing your immune system, right? What happens if you think you have liver disease? You hit that way pretty quick. Your liver also makes, you're gonna learn, your coagulation factors. Your liver makes all proteins, like it's a huge organ.
Body tissues and the fluids. The complement system comes into effect only when it is activated. Out of the 20 plasma proteins of the complement system, 11 proteins are mostly involved, named as B, C1 to C9, and D. These are mainly enzyme precursors and they catalyze a series of enzymatic reactions that form the final products of the complement system that have various immune effects. Now let's look at how the complement system is activated. The first is the complement activation resulting due to the antibody-antigen reaction, which is known as the classical pathway. The second is through the bacterial endotoxin, which is known as the alternative pathway. And the third is the lectin pathway that results due to the protein known as mannose binding lectin. Now let's study in a bit detail about the classical pathway. The main trigger of the classical pathway is the interaction between the antibodies and antigen. This reaction activates the C1 component of the complement system. The C1 component then cleaves C4 component into a smaller component C4a and a larger component C4b. The C1 combines with C4b and forms C14b. This is an enzyme which converts C2 into a C2a and C2b. These two components combine and form the C14B2A. This is an enzyme known as a C3 convertase. The C3 convertase then splits C3 into C3A and C3B. The C3B combines with the C3 convertase and forms C14B2A3B. This is also an enzyme known as the C5 convertase. The C5 convertase similarly converts C5 into C5A and C5B, out of which C5B is the largest product and combines with C6, 7, 8, and 9 to form C5B, 6, 7, 8, 9, which is the membrane attack complex. In this cascade, you can see some components like...
So does that make sense? Yeah. Now how everything is activated based on like the previous thing?
C5A and C3A, which do not have any use. They have other immune effects, which we will discuss later.
So that's what he was saying. And so it comes, you have your C2A, C2B. Okay, that A goes off right here, and same thing right here. And that's what causes your opsonization and your inflammation.
Now let's talk about the alternative pathway of the activation of the complement system. As we all just studied, the antibody-antigen reaction is the main trigger for the activation of the classical pathway. But the alternative pathway is mainly triggered by the bacterial endotoxin. The bacterial endotoxin is a lipopolysaccharide compound which is found in the outer membrane of the gram-negative bacteria. The other triggers of the alternative pathway are the yeast cell wall and the cobra venom factor. The first step is the spontaneous hydrolysis of factor C3, which is present in our blood plasma. This hydrolysis is a spontaneous process where the factor C3 is converted to factor C3b. This allows the attachment of the factor B to the factor C3b and opens the active site on factor B. The factor D, which is another complement enzyme, then splits factor B into subcomponents factor BA and factor BB. The factor BB then combines with C3b and forms the C3 convertase of the alternative pathway. This may have a different complement structure but has similar function of converting C3 into subcomponents C3a and C3b. The rest of the steps are similar to that of the classical pathway, that is the formation of C5 convertase and subsequently the formation of membrane attack complex, which has a complement formula C5b6789. Now let's talk about the lectin-mannose pathway for the activation of the complement system. The lectin-mannose pathway depends upon the protein known as mannose-binding lectin, or the MBL, which is synthesized in the liver. Now, organisms like Neisseria, Candida, and Salmonella have glucose and mannose residues in their cell wall. Whenever these organisms enter the human body, the protein mannose-binding lectin recognizes this and activates a protease called the MASP, or the mannose-binding lectin-associated serine protease. The MASP is a protease which binds mannose-binding lectin. The MASP then triggers the activation of components C4 and C2 into their subcomponents C4a and C4b, and similarly C2 into C2a and C2b. The factors C4b and C2a combine to form the C3 convertase of the lectin pathway. This C3 convertase has a similar function as described earlier, and then the pathway proceeds further as described earlier. Now let's talk about the immune effects of the various complement products. The first is the opsonization. The factor C3b strongly activates the neutrophils and macrophages. These activated neutrophils and macrophages then engulf the foreign microorganisms known as phagocytosis. The second immune effect is the cell lysis. The membrane attack complex, the C5b6789, has an effect of rupture of the bacterial cell wall, which destroys large amount of bacterial load. The third effect is chemotaxis, where factor C5a attracts the neutrophils and macrophages to the area where the antigen is present. Another complement effect is the activation of the mast cells. The factors C3a, C4a, and C5a have a direct effect of activation of the mast cells and the basophils. This causes the release of large amount of mediators of inflammation like histamine and serotonin, which potentiate the immune response. So this was all about the complement pathway. Thank you so much for watching this video. Make sure to subscribe to our YouTube channel for continual support. Stay tuned for the upcoming video.
All right, well, that's it today.